my name is Rhiannon de la Rosa. I'm a master's student with Dr. Richardson here at the University of Arkansas. I actually was also here as an undergraduate. I did high school in Iowa, and I decided that I loved horticulture and I wanted to go into horticulture. I Googled the top 5 horticulture programs and visited each school, and I just fell in love with Arkansas. Not only is Northwest Arkansas an amazing plant community, but the people here are also just fantastic. And so as an undergraduate, my research mainly focused on asparagus fern and phytoremediation, which is taking pollutants out of the air. But in my free time, I volunteered with native plant conservation communities. And so by the end of my senior year, I was actually supervising a greenhouse. And so when Dr. Richardson approached me to start this project, this Prairie Restoration project, I just was so excited. And so here I am. And so why we want to focus on tallgrass prairie restoration is that tallgrass prairie is an amazingly diverse ecosystem. It holds a lot of different animals and insect and plant diversities. However, since European settlement in America, our prairie land has gone from 25 million acres down to only 300,000 acres. That's a 99.9% loss of land. And so what we want to do with restoration is reclaim that damaged or destroyed land. Now, the way to do that is, yes, we want the animal and insect populations to return, but before we can even think about that, we need to restore the habitat. We need to go back to our roots, literally, and to restore these plant populations. The way to do that in restoration is usually by seed. This is a cost and labor saving technique, and so you just sow seed on your prairie restoration site and grow it that way. However, recently it's come into question, does it matter where the seed comes from? For example, if we have a prairie restoration in Arkansas, can we buy seed that was produced in Minnesota, or will there be negative effects? Why we believe there may be negative effects is because of local ecotypes and local adaptation. This happens because within even just one species, there are differences between populations. These populations will grow and adapt to their local habitat, and this means that they have certain traits that are specific to this population, not the entire species. And so why we're worried about this is that if we have a population of big bluestem in Arkansas that is struggling, and this population is in a wet, cool environment, if we decide to take seed from an arid grassland in Texas, we're worried that this seed, these populations, when we bring them to this wet, cold prairie in Arkansas, that they won't do as well. They're the same species, yes, but they have not grown and adapted in the same environment. And so this is why we want to kind of look at how much will this really affect the success of the restoration. And so to do that, we've been looking at 3 prairies in Arkansas. One is very far away, and that is Railroad and Downs Prairie. One is a little far away, and that is Cherokee Prairie. And one is very close to our research site, and that is Chesney Prairie. And what we'll be doing is we'll be collecting 4 plants, 2 grasses, big bluestem and Indian grass, and 2 wildflowers, which are our forbs, and those are rattlesnake master and prairie blazing star. So we're collecting 4 species from each of 3 prairies, and we're growing them all in one garden. And we do this because we don't know if these population differences we observe are different because they're growing in a different environment, or if because they are actual traits of this population. For example, if we have a population that has all red leaves, and usually our species has green leaves, we don't know if this population is red because it's a nutrient deficiency in the soil, and that's based on the environment, or if it's an actual trait that this population has evolved. And so what we do to determine if this is environmental or genetic is we take seed from this red population and seed from our normal/other populations, and we grow them in the same environment. And that's basically what we'll be doing for this experiment. We're taking seed from several different prairies, and we're going to purchase seed from retail sources to also compare that. And then we're going to grow them all in one garden and be able to see any differences. And if these differences that we observe, perhaps some grasses are taller, mature height, some may be more drought tolerant, some may be a different color. And we're looking to see if these are, 1, genetic traits, and 2, if we believe that these genetic traits will either hamper or have a positive effect on our prairies. And so as you can see, this is not a prairie, this is a greenhouse, but this is where our subjects are being grown. We will be growing our plants in the greenhouse originally, and then we will transplant them into our final garden. And then we're going to take certain physiological measurements, such as plant height, plant maturity date, flowering time, and above ground biomass. And how we'll do that is we'll cut all of our plants to the ground before winter. We'll dry all this tissue, and then we will weigh each individual biomass. And these measurements will help us estimate fitness. So if we have a very tall, heavy plant, we're going to say that that plant was more fit in this environment than our stunted small plant that didn't do very well. Now, if these two plants are from different environments, if we say that this tall plant was from our very close prairie, and the small stunted plant that didn't do very well is from our very far away prairie, then this is evidence that our prairie sourcing is very important. Because if we took this local plant and we put it in this restoration, it's going to do very well. But if we see plants that were taken and sourced from further away and they're not doing very well, then this is evidence that our restoration needs to be collected from local seed. However, if we see that all of our populations are doing relatively well, if the fitness is pretty constant throughout the board, then this is evidence that restoration seed source doesn't really matter. We can source seed from Minnesota. We can take seed from Little Rock in Arkansas and bring it up here to Fayetteville, and won't see too many negative effects. This trial will go on for 2 years, and at the end of each year, we will cut the biomass and restart the plant. This way, we'll have the same individuals and the same sources, but we'll have two measurements for any annual differences in temperature or rainfall. And so at the end of this, we will be able to determine the real importance and how far away can we feasibly get to seed source. Is it appropriate for a Fayetteville restoration to use Little Rock seed? Is it appropriate for a Fayetteville restoration to use Minnesota seed? These are all questions that we're interested in, and we believe will be very helpful for prairie restorationists in the future.